I'll just have to ask you to scroll. Great. So I'll just go ahead then. So the title is a predictive model for post-chemotherapy neurodegeneration based on RNA-Seq data. And next slide. So brain fog is one of the most commonly reported side effects of chemotherapeutic treatments. So there's CICI, that's chemotherapy-induced, that should be induced, sorry, cognitive impairment. There's also chemotherapy-induced peripheral neuropathy, and they're two different things, but they're sort of related. The first is characterized by cognitive impairment, so issues with memory, issues completing various tasks, and then peripheral neuropathy is characterized by pain generally. So what's surprising is that CICI involves the central nervous system. So the blood-brain barrier should be filtering out chemotherapeutic agents generally from having an effect on those neurons. Whereas peripheral neuropathy is more understandable given that those neurons should be directly in contact with the chemotherapeutic agent. But you see the death of neurons in both cases. So it implies that there might be an indirect mechanism. And regarding the indirect mechanism as well, it makes it difficult to study chemotherapy-induced cognitive impairment because it involves the central nervous system. So for all of those reasons, what you need is a diagnostic technique that is non-invasive. And so that's where the RNA-Seq data comes in. Yeah, I think this says everything I was just saying. So the aim of this project then is to try and build a machine learning model which can predict whether or not a patient has had a chemotherapeutic agent, and then use that model to try and identify genes that are related to neurodegeneration in order to try and build another model which can predict neurodegeneration in the case of patients who've been exposed to a chemotherapeutic agent. Yep, I think that's what that says. So the proposed methodology is use RNA-Seq data from patients who were treated with the chemotherapeutic drug and use it to train a random forest classifier. So the classifier would just look at basically pre and post treatment with the chemotherapeutic drug. The data set that I use actually had samples from patients before they were treated and samples afterwards. So it basically would just classify the sample as pre and post. And then scanning the available literature, I would look for a list of genes which have been implicated in neurodegeneration otherwise, and then look at the feature importance of those genes in my model to see whether the model basically used them to predict whether the person had been treated with the chemotherapeutic agent or not. Sorry, I haven't looked at these slides since yesterday, so I'm kind of liking what they do. All right, so everything else is in the actual notebook. Great, so just a lot of data cleaning. In the original data set, there were samples from patients before they were treated, samples during treatment, and then afterwards. So all I did was just filter out the sort of the intermediate group and just create a list of paired samples. So before treatment and then after 90 days of treatment. Yep. And scroll. Yep, just cleaning the data. Should really collapse a lot of these. And labels. All right, and then finally, the model itself. So the model was surprisingly accurate. It did a good job, actually. It correctly identified the sample around 70% of the time. And so then I just checked different train test splits. And so I found surprisingly that with the 40% split, it was up to 85% accurate. And so that's the split that I used going forward. And so then I looked at feature importance. Oh, and you can see the confusion matrix. It just, yeah, it's all fairly intuitive. So then feature importance, I basically took the top 20 features as far as importance goes, and then ran basically a quick literature review on them to see if they had been implicated in neurodegeneration. And surprisingly, about 10 of them were. And so then what I tried to do using the other literature sources that I had found was basically compile a list of other genes that should be involved in neurodegeneration and look for them in this data set, which unfortunately did not work. And the reason why is because this data set uses, there are no gene names, it's actually probe IDs, and there isn't a one-to-one correspondence between the name and the probe ID. So for like one of the genes, there's 10 different probes that corresponds to it. And unfortunately, none of them were in the data set. So that's something that I would improve on. Oh. There was one more. Yeah. And so what I did was I basically selected for those features and tried to improve the accuracy of the model. And it worked. So using 10 features out of 20,000, the model actually became more accurate. And so just scroll down a bit. Yep, and the false positive rate is actually lower.
All right, is that it?
And so there's a small part down below. Yeah, so the point of doing all this was just to identify a useful list of genes that might be used to create the model that actually predicts neurodegeneration. But for that, you would need labeled data, which I had a hard time finding.